Ladies and gentlemen, I'd like to welcome you to our online training on troubleshooting in immunoprecipitation. My name is Klaus Herig and I'm the Vice President Marketing and Sales at ChromoTek, a part of ProteinTech Group. Before we start, I'd like to quickly introduce our today's speaker, Christoph. Christoph Eckert is a marketing manager at ChromoTek. He studied biology at the Ludwig-Maximilians University in Munich and earned his doctorate from the University of Cologne. He joined ChromoTek in 2014 and gained substantial experience in the field of immunoprecipitation troubleshooting when providing technical support to our customers. After Christoph's training, we will discuss some questions that have been previously submitted. So Christoph, if you'd like to start, please.
Thank you, Klaus, for the kind introduction. Hello, everyone. Welcome to our online training, Immunoprecipitation Troubleshooting. So first of all, I would like to shortly again explain what immunoprecipitation is, and then we focus in the first part of our troubleshooting on immunoprecipitation, and we add a second part which is focusing on the troubleshooting of co-immunoprecipitation. So what is immunoprecipitation? Immunoprecipitation is a technique to isolate a protein of interest from an extract using a nanobody or an antibody. The antibody or the nanobody is coupled to a matrix like agarose or magnetic agarose. The protein of interest is then enriched on the matrix and immunoprecipitated, while unbound cellular components such as lipids or other proteins are washed away. So I would like to go through all the steps which are important in an IP now. So the first thing is you have to prepare your cell lysate. And the preparation method depends on the cell type and the protein of interest. Then you have to add a nanobody or an antibody which is bound to the matrix and incubate the nanobody together with the cell lysate for about 1 hour at 4 degrees. The beads are separated from the supernatant after incubation by a magnet or centrifugation. The beads are washed a couple of times, and the goal is to remove all unwanted and unbound molecules. The bound protein is then eluted from the beads and followed by the analysis of the eluted protein by SDS page and by Western blot analysis. So the next slide shows how a typical IP result looks like on a SDS page and Western blot. So the first, there are normally 3 fractions present. The first fraction is the input. And I would like to add that we focus in this presentation of the immunoprecipitation of a GFP-POI protein, and we use that this as an example. So the input is called IE, and the input is the total cell lysate and contains all cellular proteins and the protein of interest. Often the protein of interest can be detected in input on Western blot with an antibody, which is here the anti-GFP antibody. The second fraction is the flow-through, or is also called supernatant, and that corresponds to the input after the binding step is completed. The flow-through contains cellular proteins without the protein of interest. The SDS page shows no band, so the Western blot should be empty. The third fraction, and this is the most important fraction, this is the bound fraction. And this is obtained after elution of the protein of interest from the beads. This bound fraction contains the proteins bound to beads, and usually your protein of interest band should be visible on the SDS page and on the Western blot. So now we talked about the various steps, but also it's important to select the best antibody for your IP experiment. So a crucial part of the antibody is used that it has to work. Normally you use the antibody and it is first bound to protein A or G and then added to the cell lysate. Often antibodies are used for endogenous proteins which do not contain a common tag. That works often, but sometimes it does not work. And there are several disadvantages of conventional antibodies. One is that the antibody is eluted with SDS sample buffer from beads and is also found in the bound fraction. The antibody heavy and light chain appears then on the SDS page and on Western blot beside or next to the GFP protein of interest band. Additional antibody bands can make interpretation of IP results difficult. In addition to the antibody bands, and antibody and protein A or G can increase the background by unspecific binding. Before using an antibody, it's necessary to optimize the antibody bead ratio and the amount of antibody needed for cell lysate volume. Also, although there are many antibodies available, not every antibody works well in IP. We recommend only to use antibodies which are validated for immunoprecipitation. And then antibodies have normally a high lot-to-lot variation and may have to be optimized for each production batch again. So due to numerous disadvantages, customers use often nanobodies instead of antibodies for immunoprecipitation. I'm not sure if you are all familiar with nanobodies. Therefore, there is a short overview about nanobody or what an antibody is. On the left-hand side, you can see a mouse and below a traditional antibody containing a heavy and a light chain. The camelids, and the alpaca belongs to the camelids, they have in addition a second antibody type, a so-called heavy chain antibody. And the binding domain of a heavy chain antibody, which is highlighted here in green, is called VHH or nanobody. A nanobody is very small, just 15 kilodalton in size, and consists of a single polypeptide chain. Nanobodies can be used as research tools. When they are bound to beads, they are very often used for immunoprecipitation, such as our famous GFP trap. But what are the benefits of using nanobodies in immunoprecipitation? The nanobody is already covalently conjugated to beads, such as agarose or magnetic agarose. That means that no heavy and light chains appear on the SDS gel. They are not eluted during heating with SDS sample buffer, and the bound fraction contains only the protein of interest. No heavy and light chains appear there. Because they are covalently conjugated, the GFP trap is ready to use and no pre-incubation steps with protein A or G needed. In addition, the GFP trap has an extraordinary high affinity of 1 picomolar and can pull down even lowest amounts of proteins. Small proteins can be recombinantly produced without lot-to-lot variations you typically have when you're using antibodies. Our products are fully validated for immunoprecipitation and come along with a detailed step-by-step protocol. It's available for common tags such as GFP, mCherry, MIC, MBP, SPOT, or GST. So during this talk now, we only focus on troubleshooting for GFP tag proteins and immunoprecipitation using GFP trap, but the same principles work for other ChromoTek traps or other immunoprecipitation with conventional antibodies. So now we are coming to the troubleshooting and we are addressing first the controls. So that's really important that you conduct all the necessary controls. And then we address the questions, what should I do if I face high background and bound fraction, no pull down of GFP fusion protein, no GFP fusion protein can be detected, or what should I do if low amount of GFP fusion protein is eluted with SDS sample buffer? So the recommended controls. First of all, I need a positive control. A positive control, use the tag is just the tag only, so just the GFP. You have to test if the IP works, so you should get a positive signal in your SDS page and on the Western blot where you can detect the GFP with an anti-GFP antibody. Negative control is also very important. You do an immunoprecipitation without the tag. So in this case, no GFP should be in your immunoprecipitation. There is no fraction visible, or there is no line band visible in the bound fraction, either on SDS or in Western blot. The background is hopefully very, very slow. And then we have another negative control, control 2, but this control is optional. You can use the naked beads where the nanobody or the antibody is bound to and check if there is unspecific binding of proteins to the bead matrix. The protein of interest should not be bound. If other cellular proteins bound, naked beads can be used for pre-clearing. As a summary, controls are very important for the interpretation on OFP and for the troubleshooting. Now we move to our first problem, high background in bound fraction. So normally you should have, as I said, just the protein of interest, but often you see unspecific binding or high background in the bound fraction. You can see additional bands, and the reasons for that can be different. One might be that the concentration of the cell lysate is too high. Either too many cells are used or not enough buffer is used. Or cellular proteins, or also the protein of interest is degraded or aggregated, and the aggregates binds then unspecifically to the GFP trap. What can we do to avoid such a situation? We can focus on 3 different parts of the immunoprecipitation. On cell lysis, so you have to use the right method for cell lysis. Rule of thumb is to keep everything cool to avoid protein unfolding. And you should use the right amount of cell material per immunoprecipitation. We normally say 500 micrograms total protein, or 10 up to 7 mammalian cells, or up to 10 grams of plant material. Important is to treat the sample gentle. Keep the incubation temperature at 4 degrees and short. 1 hour is normally enough, and overnight incubation is not needed. The longer you incubate your sample with the nanotraps, the higher the background will be. Due to pre-clearing with naked beads, doing pre-clearing with naked beads to remove proteins that bind unspecifically to bead matrix. And finally, you should add protease inhibitors to all buffers, not only lysis buffer, to prevent protein degradation. In washing, you could also do a couple of optimization experiments. Add detergent or increase salt to wash away unspecifically bound proteins from beads. It is important that you choose conditions where the protein of interest is still bound to beads and only unspecifically bound proteins are washed away. For washing, it is also important to use a new tube during the last washing step. If you do not switch the tube, proteins that bind unspecifically to the tube will also be found in the bound fraction. We go now to the second case, which is also very common. No pull down of your GFP fusion protein. It's not visible in the bound fraction. Also, it's in the input. The protein of interest is not precipitated and remains in flow. Again, different reasons can explain this observation. The GFP protein is unfolded or insoluble. The GFP-Tech can't be bound. Beads are not sufficiently mixed with the cell lysate. Only a small amount of beads are used. The protein of interest is too large to enter matrix pores in case of a porous matrix like agarose is used. What can we do to avoid these kind of problems? If your protein of interest is aggregated or unfolded, it is necessary to optimize not only cell lysis, but also the expression. It's likely that your protein of interest already unfolds in the cell and not only during the cell lysis. It is important that during binding, you do a proper mixing by end-over-end rotation. Mixing by a thermomixer is often not sufficient. Always use 25 microliter of beads. If you use a smaller volume, it's difficult to pipette and to reproduce, and the amount of protein of interest can be below the SDS page detection limit. The next case we focus on is very similar to that case. The difference is that there is not only no GFP fusion protein in the bound fraction, but there is also no fusion protein in your input. So the reasons can be that the protein of interest concentration is too low because it is low abundant and below the detection limit. Or your protein of interest is insoluble because it's aggregated and found in cell debris fraction after cell lysis. What can you do to avoid this? We would recommend to increase the concentration of natively folded proteins by optimizing expression and cell lysis. Or you use more cells or a higher concentrated cell lysate to get above the detection limit. I have to add that if you cannot detect your protein in input, this is really difficult to troubleshoot, and that normally requires quite some work. The next case we would like to focus on is if the immunoprecipitation works well, but the elution with the SDS sample buffer is not efficient. You can see on the Western blot there is a faint band, but only little of the protein of interest in the bound fraction. But you can see a very, very strong band in input. So there is not really an enrichment of the protein of interest. So the explanation is that the protein of interest is still bound to the beads after elution with the sample buffer. And to resolve this issue, it is important that you use at least 50 microliters of sample buffer. You mix properly with the pipette before and after boiling. So this is now all the cases we had for the immunoprecipitation, and now we focus on the co-immunoprecipitation. So what is co-immunoprecipitation? It's essentially an IP of a GFP-bait, and together with the GFP-bait, you immunoprecipitate a second protein, the prey protein. So this is used if you would like to identify interaction partners of your GFP-bait. The GFP-bait is directly precipitated, and the bound prey proteins are then subsequently analyzed by Western blot or by mass spectrometry. Again, I would like to make a short introduction on controls, and then we add 3 different problems: no pull down of the GFP-bait, no pull down of prey, and unspecific pull down of prey. So if we talk about co-immunoprecipitation, we have to talk about controls. These are absolutely important to confirm your results and to see if there is a problem, where the problem is. The co-immunoprecipitations are usually analyzed on Western blots and not on SDS page. In our example, we use, again, antibodies against TAG GFP and an antibody against prey. Often a loading control is added, for example, GFP-POI, but we don't show that here. So what can we see here? We have here in faint green our positive control. The positive control shows on the left-hand side the input, and then on the right side is the bound fraction, which is called here IPGFP. So we first would like to check if our immunoprecipitation really works, and we add therefore one fraction the GFP, and in another experiment the GFP-bait. And after immunoprecipitation, you can confirm that the GFP band is there in one experiment, the GFP-bait band is there in the other experiment. The second control we would like to include is the negative control. The negative control is once the prey, and in the second experiment, prey and GFP. In input, you can see one prey band, and in this other experiment, a prey and a GFP band. If you conduct an immunoprecipitation, then you see in the bound fraction no band for the prey protein, and you can see just a band for the GFP protein and not for the prey protein. And then if you conduct the proper co-immunoprecipitation, co-immunoprecipitation experiment, which is here in bright green. You have in the input the GFP-bait protein and the prey protein. And if you do an IP, both proteins must be detectable by Western blot, in the Western blot by the prey antibody and the GFP antibody. This is how a really good working co-immunoprecipitation looks like. And if you achieve that, I can really just say congratulations. So we come now to our next case. What happens if we face a problem? In this case, there is no pull down of the GFP-bait. The input looks essentially the same as expected. You can detect the GFP protein, you can detect the GFP-bait protein, the prey protein, and so on. But if we look to the bound fractions, we can see that the GFP immunoprecipitation worked well, but there is no band visible in the red circles. So the GFP-bait band, which should be present there, is not visible in the positive control. The negative control, again, looks good. But if you look in the proper co-immunoprecipitation experiment, you cannot see prey and you cannot see a band of the GFP-bait. The solution is to see why the GFP-bait immunoprecipitation does not work. Maybe the GFP-bait is not soluble, or just smaller bounds of the GFP-bait are precipitated. So we really have to see what the problem is, and we really focus in this case on the bait protein. As we discussed previously, in the case no pull down of GFP protein of interest, you have to look if the protein expression is working well. You have to check your cell lysis, and you have to optimize your immunoprecipitation. You have to do a proper mixing of the solutions. You have to test different matrices. You have to check if you really use 25 microliters of beet slurry. So, but essentially is you have to see that the immunoprecipitation of the GFP-bait is working. The second case is that we look to the immunoprecipitation of prey, and there is no prey visible. So what's the difference before? Again, the input looks really good. The positive control, the negative control, and also the input of the proper experiment shows the pattern, the expected pattern. Also, the positive control looks good. You see a GFP and a GFP-bait band there. Also, the negative control looks really well. But what you do not see in your proper co-immunoprecipitation band is the prey protein. So why is there no prey protein? One reason could be that the prey protein is not soluble or unfolded. Maybe it's also not efficiently precipitated, and you can see only very small amount, or you precipitate only small amounts which are below the detection level. So the solution is you have to optimize the conditions for prey. You have to test different cell lysis buffers and binding buffers. You should also consider whether there is really an interaction between bait and prey. It's also possible that if you use a bait protein with a tag, that the tag interferes or prohibits the interaction. So that's really tricky, but really focus if with prey, the expression of prey and everything is okay. The next problem is if we have an unspecific pull down of prey. Again, the prey input, or the input looks really good. But you can see that in the negative control, you can see two bands which should not be there. And this is an indication that you have an unspecific pull-down of prey. And here you can see how important controls are, because if you look at proper experiment, you see the prey and the GFP-bait band. So the proper co-immunoprecipitation results look really nice. But when you have a really careful look to your negative control, you can see these additional bands. And this really indicates, or is it not indicates, this is a proof that somehow your results cannot be trusted. Why is there prey protein detectable in the negative control? We assume that normally prey should be unfolded or aggregated so that it binds to the beads in a non-specific way. The solution, again, you have to optimize the conditions for prey. Test different cell lysis buffer and binding buffers. Do these tests without the GFP-bait. Just optimize the conditions for prey. And once you found good conditions, then you have to make the whole test together with the GFP-bait protein. So this is a really good example how important proper controls are. So I would like to summarize now the troubleshooting of this online training. Very important, choose an appropriate nanobody or antibody for your immunoprecipitation. Proper controls are really essential for meaningful results, and controls are really important if you would like to do a proper troubleshooting. Immunoprecipitation, especially co-immunoprecipitation, requires a lot of optimization and is not always easy. Thank you very much for joining our online training about immunoprecipitation troubleshooting. Thank you very much.
Yeah, thank you, Christoph, for your in-depth introduction into the trouble that may occur during an immunoprecipitation and a co-IP. We will now switch over to a questions and answer session. We have selected 4 questions that have already been submitted previously. And we would like to start with the first question, which is, when I elute my glycine buffer, the elution is still incomplete. What can I do? So, Christoph.
So we could already prepare some slides as these questions have been submitted in advance. And as you can see here, you see a very strong band in the bound fraction, but a faint band in the elution fraction. That means that most of the GFP protein remains bound to the beads. So the majority of the GFP protein remain bound to the bead because the elution is not really effective. What we recommend is mix the beads with a pipette for 60 seconds during elution, and then repeat the elution. So you should prepare more than two elution fractions, and usually the second fraction contains most of the GFP fusion protein which you are interested or which you are looking for.
Thank you, Christoph. Next problem is, I still see my protein of interest in the flow through after binding. What's the reason, and how can I improve the binding efficiency of my pull down?
So the GFP fusion protein is seen in input, flow through, and bound. And in flow through, you should normally not see any band. So there are 2 reasons for that one. One might be that the GFP fusion protein is present in higher amounts than the binding capacity of your nanobody or your antibody is. The second thing is that maybe a fraction of the GFP fusion protein is unfolded or aggregated. What we recommend is you should use more beads or less cell lysate. You should really mix properly, and the optimization of protein expression and cell lysis conditions is really good. A longer incubation time usually does not really help to increase the efficiency of an immunoprecipitation.
Thank you, Christoph. The last question submitted was, what should I do if my antibody binds unspecifically?
So what should I do if my antibody binds unspecifically? There are a couple of problems which may arise. One is, for example, that you have a very high background in your bound fractions, and you do a lot of troubleshooting, and you do not really achieve any optimization. I would really focus on the antibody and test if there is another antibody which does not bind other cellular proteins unspecifically. Maybe you could also, if you're working with an endogenous protein, you could also add a tag such as GFP, MIC, V5, or SPOT to your protein of interest and use another reagent. For protein tags or peptide tags, often validated antibodies or nanobodies are available. And use only reagents validated for immunoprecipitations and reliable suppliers. Last but not least, if all this troubleshooting fails, you may think to adapt your experimental setup. That's then the last thing you might consider.
Thank you, Christoph. Again, so this was the last question that was submitted, and I'd like to thank everybody for your attention. Thank you, Christoph, again, and would like to close the online training now.
Thank you very much. Have a nice day. Bye.